Hi, my name is Benjamin Calado.
Hi, my name is Samuel Angelo D. Regalado.
My name is Arman Relucio.
Hello everyone, I'm Teddy Ray, and today we're gonna discuss about DNA cloning.
Reyes, Gerald Randall Reyes.
My name is Mark T. Recalde.
Cloning adult animals.
The word cloning means making an identical copy of something. It can also refer to tampering with the reproduction to produce an exact genetic copy of an organism. Reproductive cloning is any use of several technologies that produce genetically identical individuals. Differentiation. It is a process in which cells become specialized during development. Occurs as different cells in an embryo begin to use different subsets of their DNA. SCNT, somatic cell nuclear transfer, is a reproductive cloning method in which DNA of an adult donor's body is transferred into an unfertilized egg.
SCNT is now a common practice among people who breed prized livestock. Among other benefits, many more offspring can be produced in a given time frame by cloning than by traditional breeding methods. Cloned animals have the same championship features as their DNA donors. Offspring can also be produced from a donor animal that is castrated or even dead.
As the techniques become routine, cloning humans is no longer only within the realm of science fiction. SCNT is already being used to produce human embryos for medical purposes, a practice called therapeutic cloning. Undifferentiated stem cells taken from the cloned human embryos are used to treat human patients and to study human diseases. For example, embryos created using cells from people with genetic heart defects are allowing researchers to study how the defect causes developing heart cells to malfunction. Such research may ultimately lead to treatments for people who suffer from diseases that are otherwise incurable. Human cloning is not the intent of the research, but if it were, SCNT would indeed be the first step toward that end. Cloning DNA.
DNA cloning starts by having a single or double-stranded DNA that contains the specific DNA sequence or a piece of strand that you wish to duplicate or to clone. In order to cut the specific nucleotide sequence, we use a restriction enzyme to cut that strand. The restriction enzyme recognizes a specific base in DNA form. The enzyme cuts the DNA, leaving a single-stranded tails where it cuts the DNA. The DNA strand that was cut is placed in a plasmid, a piece of genetic material that can replicate independently of the chromosomes. The single-stranded tails, or also called as sticky ends, connects the two DNA fragments when their matching tails base pair. The enzyme DNA ligase can be used to seal the gaps between the base paired sticky ends, so continuous DNA strand may form. Basically, through cutting the DNA, we use the restriction enzyme, while for pasting the DNA, we use the enzyme DNA ligase. The result is called recombinant DNA, a hybrid molecule that consists of genetic materials from two or more organisms.
The first step in DNA cloning is making a recombinant DNA. DNA cloning is a set of laboratory methods that uses living cells to mass-produce specific DNA fragments. A DNA fragment is inserted into a cloning vector to carry foreign DNA to host cells. A bacterial plasmid is an example of a cloning vector. A plasmid that carries a fragment of foreign DNA can copy and distribute to descendant cells along with the plasmid's DNA. Such cloning vectors are cultured in the laboratory with the host cells to obtain a huge population of genetically identical cells or clones. Each clone contains a copy of the vector and the inserted DNA fragment.
cDNA cloning.
In cDNA cloning or complementary DNA, DNA contains introns. Unless you are eukaryotic cell, it is not very easy to determine which part of eukaryotic DNA encode gene diagram. The researchers who study gene expression in eukaryotes often start with mature mRNA or messenger RNA. Post-transcriptional processing removes introns from an mRNA, so just the coding sequence remains. An mRNA cannot be cut with restriction enzymes or pasted with DNA because these enzymes work only on double-stranded DNA. Cloning with mRNA requires reverse transcriptase, a replication enzyme that uses an RNA template to assemble a strand of complementary DNA or cDNA. DNA polymerase is used to copy the cDNA into a second strand of DNA. The outcome is a double-stranded DNA version of the original mRNA. Like any other double-stranded DNA, these fragments may be cut with restriction enzymes and pasted into a cloning vector using DNA ligase.
Now it's time for some questions. We know that you can do it. Thank you for watching.